Good morning, my name is Ketty Patel, and today I'm going to talk about gene therapy comes of age. Gene therapies are becoming a very critical component for the therapeutical collections for a variety of inherited and acquired human disease. In this article, we are going to talk about the development of gene therapies from genetic engineering from retroviral vector to genome editing. So recombinant replication defective viral vector was the first molecular tool that were used to transfer gene into the somatic cell. Retroviral vector and adenoviral vector has shown promising results. Identification of cell signaling and creating a producer cell line are important tool to design a vector that can undergo reverse transcriptase and DNA integration, but lacking of replication potential. Gamma viral vector were developed, and they was the used to transfer the or deliver the gene into the repopulating hematopoietic stem cells. Later, C type of viral vector were developed and had efficiently transfer gene into the T lymphocytes. Later on, two other types of retroviral vector was added: lentinoviral vector and spumaviral vector. Lentinoviral vector can transfer gene into non-dividing cell. Also, it can carry larger and complex genome cassette. So in recent studies, the all HSC applications, lentiviral vectors are widely used. The removal of endogenous strong enhancer elements using self-activating or SIN design can be another approach in gene therapy to reduce the risk of genotoxicity. Adeno-associated viral vector were engineered from the non-pathogenic and paravirus, where all the viral genetic sequence were replaced with the gene of interest. The limit factor for this vector is cannot package the DNA more than 5 kb. Adenoviral vectors are very predominantly non-integrated, meaning the transcribed DNA is stabilized as an episome. This factor reduce the risk related to integration, but also limit the long-term expression of adenoviral vector to cell. Gene editing technologies is a contrast to viral vector, where using genome editing techniques, we can do gene editing, gene ablation, or gene corrections. Gene editing can be done by two ways: in vivo and ex vivo method. In vivo method, editing machinery can be introduced into the body. The target DNA alteration can be done by creating a nucleus which induce the double-stranded break and stimulating the highly efficiently recombinations in mammalian cells. It can be done by using non-homologous enjoining method or homology direct repair method. You can alter the sequence of interest, whatever the gene of interest. So in earlier genome editing technologies were specific engineered zinc finger nucleus or a mal nucleus, and later on the discovery of tail nucleus, its enzyme has capacity which can efficiently cleave the DNA of any interest. Later on, scientists show that a bacterial genome consists CRISPR, meaning repeatedly integrated short palindromic region. Using CRISPR-Cas9 is an enzyme has a highly efficient to reprogram to cleave the DNA at the site of interest by using simply creating a guide RNA, which is a complementary sequence which is complementary to the target of the site sequence. CRISPR-Cas9 mainly rapidly developed and make a genome editing technique very simple. Study shows in gene therapy using in vivo method, adeno-associated viral vector were used for the clinical trials for the hemophilia A, hemophilia B, and some inherited blindness disorders. Also, it was used to study for the neurodegenerative disorders. Gene therapy using ex vivo method, in which the um, gene modification can be done outside of the body, and then once the therapeutic factor achieved, it can subsequently transfer back into the body. CAR therapy. So, engineered T cells are a very powerful tool or medicine for the cancer. Chimeric antigen receptors are synthetically engineered receptors for antigen. It can transfer into the T cell via ex vivo method. After that, the new generation of CAR T cell require very stable gene transfer, and it can be done by using a gamma viral vector, gamma retroviral vector. They were first used where to express CAR expression on CD19 is, my opinion, the gene therapy is the most complex drug ever developed. If but more involvement of biotechnology and pharmaceutical companies, we can create more and efficient gene therapies for those who are suffering from very serious human disease. But even though we have to face the challenges related to genotoxicity, long-term cell gene expression, and immune response. Thank you.